Smoky mice are interesting for many reasons. They're cute, they're fuzzy, they smell nice actually, but that's not at all the reasons why I'm interested in them. I'm really interested because they're enigmatic and they're part of an amazing diversification of species that occurred in Australia once rodents arrived 4 million years ago. They're also a member of a group heavily impacted by extinction since the arrival of Europeans, with more than 40% of all mammalian extinctions in Australia being rodents. Smoky mice are one of the survivors, but they're also an endangered species threatened with extinction. When you lose genetic diversity very rapidly, that can lead to new diseases and reduced health of populations. So in the Grampians garroward, for instance, we only know them from about 6 kilometers along the Victoria Range and along about 6 kilometers the Mount William Range. And we've now collected genetic samples and documented and tracked more than 150 individuals over the last decade. The traditional way that we collect samples without sacrificing the animal is we take a small ear biopsy, a little sliver from the edge of their ear, typically stored in some sort of preservative that preserves the DNA. But that DNA is dead, and more recently, because we're trying to preserve the genetic diversity that's living in populations today, we now use techniques that preserve the living cells from that same ear snip, so that someday we might be able to use those living cells to reintroduce genetic variation into the wild. Either we have to keep the cells alive on ice and get them culturing and growing in facilities within about a week. The other method that we've developed and tested with smoky mice is to flash freeze the samples in liquid nitrogen, which is not that easy a task because if you just freeze them, they'll explode and they'll rupture. But we put cryoprotectants in there and we can flash freeze those cells in liquid nitrogen, which means we have to carry in a liquid nitrogen tube on a pack, carry that over fallen logs and over big heavy boulders. And those samples can stay frozen indefinitely, we think. We're testing that now. We brought them back, we've held them for months, and then we've been able to grow them in our facility at Melbourne Museum. That little bit of ear contains skin cells, and we bring that back and take a little piece of that and score it into a petri dish with a cell culture medium, a medium that those cells love to eat with all the food and nutrients they need to grow. And then over a matter of a week to 4 weeks, they grow in a low oxygen incubator. So once we've grown the cells to sufficient quantities, many millions of cells, we can sample them and then store them. We put them into one of our cryotubes and then slow cool them down to liquid nitrogen temperature. That's minus 196 degrees Celsius. At that temperature, they're basically cryopreserved alive in stasis. And we can pull them out at a later date and thaw them out and then grow those cells again. We can do studies on those cells, or we can use those cells to modify and reuse in the wild. In this case, we're not able to get reproductive material, sperm or eggs from smoky mice without harming them or great difficulty. And now we're just collecting body cells, what we call somatic cells. How do you put those back into the wild? And current cell biology is at that point. So you can take a cell and you can reprogram it to become a pluripotent cell. So pluripotent stem cells are cells that can turn into any other cells. We all started from a single cell and a fertilized egg, and that became all our different body parts, all our different tissues. We can now do that in lab animals. And it's essentially the technology that the Thylacine de-extinction project is attempting to do. With the thylacine, though, they have to go to the great extent of then editing that genome. And they know the thylacine genome from our collections here at Museums Victoria that store the best DNA of any thylacines. That's part of what's motivated us to say, we need to put away live cells, not just dead DNA, because look at the efforts we're going to try to recover the dead DNA of a thylacine that was collected 113 years ago. And it would be so much easier if we started with living cells of that animal. Museums for the last 3 centuries have documented biodiversity, species diversity, and the genetic variation within species. And that record tells us that we live on a rich, living planet, but it also tells us that diversity has declined. And we know that there's unique genetic variation stored in these cabinets and these collections here that is no longer present in living animals on Earth. And we have to ask ourselves what's present today that we're going to wish we could have living out in nature that's housed in our collections. And thanks to our efforts with cryopreservation of living cells, we're going to have an increasing number of our specimens that document the genetic diversity of today preserved with living material for the future.